Let's say we have a chromosome, and when unraveled, a chromosome is one long string of DNA with many different genes on it. Each gene codes for a different protein. For example, you might have several enzymes in a single stretch of DNA. Now, typically, every gene is going to be preceded by the promoter region, and it's going to be capped off by the terminator region. But sometimes you need multiple enzymes for the same task. For example, when we were talking about glycolysis, you may remember that it took 10 or 12 enzymes in order to go from glucose through that full metabolic pathway in order to produce pyruvate. So there are times when the cell might want to produce all of these enzymes simultaneously, particularly if it doesn't need them all the time. When the need arises, E. coli bacteria are able to digest lactose in order to gain energy. Lactose is a disaccharide made by creating a glycosidic bond between a glucose and a galactose monosaccharide. Lactose is fairly rare in E. coli's environment, but when it comes across some lactose, it would like to use it for food, so it's going to need to transcribe all of the enzymes required to break down that lactose at the same time. In order to accomplish this, it strings them together in what we call an operon, one after the other, sharing a common promoter region, such that when RNA polymerase transcribes one of these genes, it transcribes all of these genes. Now, building up proteins requires a lot of energy and work, so the E. coli bacteria isn't going to do this all the time. It's only going to do it when lactose is present. So it puts a kind of padlock on the gene. There's a section called the operator, and attached to that operator is another molecule, another protein, which is called a repressor. That's our padlock. And this padlock has a key, and the key is the lactose molecule itself. And in this way, E. coli bacteria only produce the enzymes for digesting lactose when lactose is present. This is an example of an operon, which is just one way in which cells can regulate gene expression.